Prashanti madam. Prashanti madam.
Dhatri?
Section start చేసేయండి మేడం. సార్ ఆల్రెడీ జాయిన్ అయిపోయారు.
Okay. Good afternoon to everyone. Welcome back to third session of AICTE ISTE refresher program on role of artificial intelligence in drug discovery and development, which is organized by VV Institute of Pharmaceutical Sciences. I hope you have experienced two excellent sessions in the morning, and you will also impress with this session also. And it gives me immense pleasure to introduce present speaker, Dr. P. Ramalingam, Associate Professor in Pharmaceutical Analysis from Naipar Institute. Sir completed his B.Pharmacy in the year of 2000 from RVS College of Pharmaceutical Sciences, Coimbatore, Tamil Nadu. And he completed his post-graduation in pharmaceutical chemistry in 2003 from College of Pharmacy, SRIPMS, Coimbatore, Tamil Nadu. And he secured first place in his PG. Sir was awarded with PhD in Pharmaceutical Sciences during the year 2011 from Andhra University, Visakhapatnam. He had completed CMI Level 5 in Management and Leadership AICTE UK IERI Scheme 2019 by Chartered Management Institute, UK. Sir started his career as assistant professor in pharmaceutical chemistry in 2003 and worked for many colleges and gained over 17 years of teaching experience. Now he is currently working as associate professor at National Institute of Pharmaceutical Education and Research, Hazipur, Bihar. Sir was guiding 7 doctorate students and supervised 52 PG students. He had organized over 11 conferences and also delivered so many talks, so many guest lectures in many events. Sir had also 31 research publications in national and international Scopus indexed journals. Sir received about 1 crore research grants from various institutions like National Institute of Research in Tuberculosis, AICTE, etc. Sir also acted as a project coordinator for DST FICT 2017 Government of India. He had completed 2 consultancy projects from Waxman Selman Pharmaceutical Private Limited and Atal Incubation Center, SK University, Anantapur. Sir published a textbook on application of spectral studies in pharmaceutical product development, and he also written 4 chapters in various books. Sir was a founder editor of iCare e-News Bulletin and also an associate editor of Riper PDIC Bulletin, and he is a life member of IPA and APTI. Sir also received many prestigious awards and have many recognitions. In that, he received University Best Teacher Award in the year of 2021 by JNT University, Anantapur, Andhra Pradesh. He got BIRAC GYTI Appreciation Award 2016 for his research by TBT Government of India and also secured a cash prize of 1 lakh. He got Best Teacher Award 2016 by Indian Pharmaceutical Association, Anantapur Branch, India. He had received certificate of appreciation from the government of Andhra Pradesh, India for being coordinator of online GPAT classes for pharmacy students organized by Department of Higher Education, Government of EP. He is president of IPA local branch and received national award in 2019 as a best local branch. He trained and certified as evaluator for National Board of Accreditation visits by NBA Nodal Center, JNT University, Anantapur, India. He was a Central Council member of IPA Education Division from 2017 to 2020. He was an enlisted medicinal chemist in anti-tubercular drug discovery supported by TAACF, US National Institute of Allergy and Infectious Disease from 2006 to 2009. He is a member of Board of Studies, JNT University, Anantapur. He is visiting faculty under UGC Visiting Professor Scheme to Acharya Nagarjuna University, Guntur. He is a doctoral advisory committee member and recognized research supervisor of JNTUA, MAHE, JSSAHV. And so also received 2 patents, one on use of gum produced by Gummosis of Ennona species as phytochemical excipients, Indian patent, and one is on efficacy enhancement of quinone molecules in combination with isoniazid and rifampicin. With his inspiring profile, I cordially invite to deliver his talk on computational approach in the prediction of analytical behavior of analytes. Sir, welcome, sir. Sir, Ramalingam, sir.
Yeah, am I audible?
Yes, sir. You are audible.
Okay. Very good afternoon, and to BBIIPS and principal and organizing committee members of this program, which is funded by ISTE. And at the outset, I thank the principal Lakshman Rao and the team of faculty members and advisory members of this event for inviting me to deliver a session on this computational approach in analytical method development generally. So, with this and without further delay, I wish to. Can I share my slide? Is it visible?
Yes, sir.
Ramalingam sir, if you don't mind, can you switch on your video for a while?
Yeah. I could not switch on the video. That's the problem here. Just
Okay,
very, it's in default. I think somewhere it's a problem here.
okay, sir.
Just wait.
No problem.
No. Actually, by default, it's switched off. Actually, I switched on. From your end, can you check whether you can switch on my video?
Sir, it is default switched off, sir. Actually, the camera is not found here. I think that application is there is a problem in the
No.
application.
First, when I share my video, it was there, right? Just wait. Can I leave and join back?
Sure, sir. You
Sure,
can.
sir.
Is it not a problem for you, right?
Yes,
Just a
sir.
minute.
No
Yes,
problem.
sir. Yes.
Okay.
Dhatri.
మేడం
ఓకేనా అంతా?
ఓకే మ్యామ్ అంటే ఒక్కసారి ఫోటో తీసుకుందామని చెప్పాను.
ఓకే, ఓకే.
I think right now is fine.
No, sir.
Video is not.
Yes,
No.
sir.
Yes, sir.
Yes. Yes, sir.
Okay. Okay. The slides are audible. I'm visible.
Yes,
Voice.
sir.
Okay.
Sir, you are not visible.
I don't know when. No. The problem is when I share my screen and automatically my camera gets switched off. I don't know.
Sir, can you stop your screen share for a while? I just take a picture of you.
Okay. Just wait. Okay. Now, right?
Yes, sir.
Okay.
Thank you, sir.
Okay then. Thank you. I think. Hope my voice is audible and screen is visible.
Yes, sir. All are perfect, sir.
Okay then. Okay. I think I will start now, right? Okay.
Yes, sir. Yes.
Okay. Good afternoon once again to all the participants and delegates from the different port of entry on this online platform. And it's my pleasure to interact with you and deliver a session and on computational approach in the prediction of analytical behavior of analyte. So before begin my detailed lecture, warm greetings from Naipar Ajipur. So these are the content I would like to discuss throughout the presentations. They about the computational approach in analytical methods, prediction of spectrograms, prediction of retention of analytes in chromatography, and quantitative structure retention relationship, which is called as QSRR, analytical quality by design, abbreviated as AQBD, and also something about chemometric methods. So let us come for before going for the detailed topic. I wish to discuss you or bring to your knowledge about this computational approach in analytical methods, where we are, where we stand. You're aware of that the technology is day by day, it's advancing like anything. Things are moving ahead. The technology advancement is really it is uh, the slope of technological advancement is very significantly. It's growing like anything. You can see the best example: 1G, 2G, 3G, 4G. Now it's a 5G. Even you can see in the many of the computer related things, right, including your mobile and your applications, etc., etc. It's all growing like anything. So this technology advancement, the computational and the computer and software related approach, and especially the artificial intelligence in computational methods. So these are not only included in the technology like engineering or something else, electronics. It is also in the pharmaceutical research. And if you look at that, even in the recent time for the past 10 years, and if one decade, if you assess, if you go back and assess, you can see that many in silico based approaches, computational approaches, are in drug development. Notably, you can say that in silico drug discovery, the tools like molecular talking, virtual talking, de novo drug design, and you can also say simulations, molecular dynamic simulations, and the many are in contributed significantly in the drug discovery, and especially if you take that example, the cimetidine H2 blockers and many antivirals, DNA gyrase inhibitors, topoisomerase inhibitor. The many drugs have been discovered by using this in silico approach, like molecular docking and MD simulations. And also now even FDA has approved many of the software to be used in toxicity prediction. Recent time the toxicity also very important concern, yeah, for a drug discovery scientist. And around 10 years or to decade ago, the scientists who work in a drug discovery, they focus only on the potency and efficacy. They always keep this toxicity at the later stage, and they focus. Now it is not the era now, and whomever they are involving drug discovery research, it is very very essential that you should focus on all three equally. When you design a molecule, when you screen a molecule against either in vitro or in vivo studies, the importance has to be given, like a toxicity prediction. The toxicity of the molecule is also very essential. So here the even the toxicity prediction software tools, in silico tools are available, the same that of molecular docking and MD simulation for everything. So these are well known in drug discovery area. Also, when you come for your clinical trials and in the pharmacokinetic and BA/BK studies, so in all areas, even the in vitro in vivo correlations in pharmacokinetics and everything, like the calculations and prediction of pharmacokinetic parameters for a drug, and everything is the tools are available which are developed based on artificial intelligence. They are coming under computational approach. In addition, that there are many tools and many softwares are day by day they're developing for predicting the drug interactions, drug informations, as well as the many software has been used and computational tools are introduced as diagnostic tool. The very simple example: if somebody goes to hospital, they record a ECG, and the ECG, and before you go to a doctor, you can see that the ECG result impression, the machine itself can give the possible myocardial infarctions. It's a normal ECG, or possible supraventricular atrial fibrillations. So likewise, you can also see that particular comment in the ECG machine, the diagnostics, it is somewhere around 20 years ago only, this computational approach in the prediction of data, diagnostic data is being introduced. So likewise, it is not the computational approach in our health sector or pharmaceutical sector is not new, but when I want to stress, unlike this drug discovery area or in case of pharmacokinetics or anywhere else, and this validity of computational approach in analytical chemistry and yet to be assured. So there's use of prediction tool in analytical chemistry, especially why we use this prediction tool in analytical chemistry to predict the behavior of analyte. Because especially in India or some country where we have the resource limited setup, and people, once upon times, so, a compound is there, and I'll give you example. Yes, synthesized on chemical compound, and if you want to see how this particular compound show the IR spectrum or Raman spectrum, until unless you send the compound to some laboratory, you pay some around 100 or 200 rupees, you get the spectra, then we'll understand that how this molecule is showing its pattern, and how this IR spectra appear for this compound. Now, this is changed. Things are changed now. In online, there are many free softwares are available in the internet, and only concern to the the kind of, I don't want to make it as advertisement for some of the software, so I have not mentioned the name of the software in the slide for this reason. And there are, you can search that there are many softwares are available in the online, and where you just give a structure that can show you the possible IR spectra. It will show that how NMR spectrum will be there, how IR spectrum is there. So there are tools are available, how UV spectrum will come, what would be the lambda max. So all the things are, it's available in the current internet and the computer era. And but only thing is that what I want to say, that the validity of this computational techniques and analytical chemistry, especially for prediction, is yet to be assured. So why, in my opinion, and the computational approach in the analytical research is equally important, and it can also provide the simulations, right? These particular, what are the simulations we are going to do, that can provide a vital insight into chemical behavior, and also it will allows for hands-on modeling experience, and also it will provide the visual representation of the correlation of theory and real experiment. That's what here it's very important, that until and unless, that's also always true. So, despite the technology advancement, though you are able to predict everything with a less percentage error, until and unless you go for your real experiment, and the computational tool will not be valid. So that's what I made a statement that, and everywhere, even in drug discovery also, you bring out some lead on promising molecule that can significantly pose with your protein. It has a very good glide score or docking score, or it has very good, molecular dynamic simulation result. The druggability is assured. But until and unless you go for a real experiment, it is not true. So here, one thing I want to insist to all the researchers, that don't think that these, whichever may be the area, the computational tool which you are going to use in your research, either in your drug discovery or analytical, they're all just a prediction tool. It will give you a basic idea. It is not the endpoint. It is not the end point. Don't think that we can do our research, and being a postgraduate students or a UG student, just to we are used some chemical structure, we predicted a postgraduate score. That is only, it is only 20% done, 20 to 30% done. That will give you a basic idea. As I told you, that it's a kind of, it will give you a modeling experience, and it will act as a self-learning tool. That's it. Like, it's a kind of prediction. So until and unless, it's very important that whomever may be this, it is a common statement, whomever they do your computational tool in your drug discovery or your analytical method, the prediction is only half your way. And after prediction, you do your real experiment, and you try to correlate the real experiment with your computational prediction. That will provide you the validity. That's very important. So the real experiment is going to be an endpoint. This computational approach, anywhere, it is not the endpoint. So here, until and unless you have a real experiment, you cannot validate any of your prediction, any of your computational approach. And so here, it's very important that when you come for a drug discovery, so in view of your our analytical approach, And spectroscopy and chromatography, these two approaches, the instrumental analytical approaches, are playing a vital role in especially purifications, identifications, and characterization of the target chemical compound. That is very true. Nobody can deny that without spectroscopy and chromatography. And a purification and identification characterization cannot be done in general. And if I speak generally, let us take an example of your resource limited setup. And like a institution or the student, they're studying in a remote area. The institution is not having access to the potential researcher for guidance, or the institutions do not have any adequate facilities. In such a case, basically the interpreting NMR, UV visible, IR spectrograms, and this chromatography, the understanding that retention of the chromatography, these are really difficult for the beginners. Not only for beginners, and though people are having experience, even for the non-chemist people who are handling this spectroscopy and chromatography also, it's quite difficult to interpret exactly at the molecular level or the physical chemistry level. But always it is essential that the knowledge on the spectrogram and chromatography is very essential for researcher in drug research. So that's what indeed the awareness on the computational tools will partially assist the researchers for validity of their analytical data. So here I'll tell you a case example that how you scholars or teachers can use this computational tool in your research. And apart from that, which is not there in the slide, I would like to share with you all that, for example, now in India, it is the mushrooms of predatory journals. Just like that, they publish the data without peer review. The data which is published are overnight. That's why they called us. There are all the predatory journals. They are ruining the complete scientific knowledge and scientific validity. So here, and if at all, for example, you are going to start your research, and any research you start doing, you require a literature. So when you go through a literature, that there is a literature and you are reading out, there are some analytical data available like related to spectroscopy and chromatography or retention, and they are showing that this particular molecule from the natural product shown retention on this particular column at this RT. So here, and this computation tool you can use to validate that available literature, whether it is real data or it is a fabricated. That is true. So you take any paper from the predatory journal, take those data which are available in those journal, you just same data, you just put it into the computational tools in prediction. You can correlate that predicted with the available data in the paper. So such a way that you can ensure that the data available in the journal, whether it is also true or it is how far it is true, not 100%, at least 80% you can assure the validity of the particular data available in the literature. This is a way, one way we can use it. So now when I come for this prediction tools available for spectroscopy, and no doubt the prediction tools are available for UV visible, infrared, Raman spectroscopy, and nuclear magnetic resonance spectroscopy, and mass spectrometry. So it's all the prediction tools are available for all the spectral or most of the spectroscopy techniques. And similarly, computational prediction of retention time in chromatography. For that also, the many computational approaches have been introduced. There are many papers were published in the literature that are of one. The very important one is the artificial neural network based. It's a kind of I mean, I just wanted to say that most of the computational prediction all based on artificial intelligence. Among that, this artificial neural network, response surface methodology, design of experiment, analytical quality by design, chemometrics, and the quantitative structure retention relationship. So here, as I told you that amongst all, artificial neural network based programs are widely available these days. So now I'll go into this, the prediction of how generally based and this whatever the data available, whatever the data I'm presenting, they're all data generated by us. We have taken some structure, we have predicted, and we have some published data. We try to correlate it, and that data only I'm presenting here for your kind knowledge. So whatever the in case of prediction, mostly this prediction of NMR and I mean both C13 NMR as well as protein NMR, and these softwares are purely developed by using artificial intelligence only. The examples of software available are, Kendra, everybody aware of this, and Chemaxon. They're all things you can able to predict the C13 NMR as well as protein NMR. So here, for example, and you are a chemist or a medicinal chemistry student or a cognitive student, and you have a new compound, and you are sent for NMR, and the spectra has come. And how to validate that? What you can do is you can use these tools. You enter the structure and predict the signal. It will give you that how many NMR signal can give for the structure, and for each signal, and what is the probable chemical shift value. So all those information will be provided by these tools. So that can be correlate to your real spectra what you get. That is fine. So usually when you look at this artificial intelligence for this C13 and H1 spectra, and usually as of now to the literature, and there are two approaches they follow. One is the quantum chemical calculation based method. The another is the machine learning approach in NMR prediction. These are two over, but out of this, and the first one is not that of accurate, but this approach, the software or the computational tools which follow and which is working on the principle of this machine learning approach, and especially this called as automatic structure verification, called as ASV. So this can visualize the NMR signal for the given structure more accurately. So here, for example, that it is the information finally why I am speaking all this software, all those cases, thing. But so of course we are the pharmacy people, we are not software engineers. So what I say that there are plenty numbers of softwares are available to predict this NMR spectra for the given chemical compound. I wanted to say that when you use a particular software tool, you just ensure that the I'm saying the validity of the data, how far it is suitable. So as I told you that there are two kind of approach they're designed. So you can choose any software which is working at the principle of machine learning approach. So that can predict the NMR signal and parameter more accurately. And moreover, one of the very interesting and advantage of this particular machine learning approach based NMR prediction, the computational approach is, and they have made it in such a way that the prediction will consider the temperature, solvent, pH, concentration, salt content, etc., etc. All the interference and the effect of those interference on the shielding and deshielding of the particular proton has also been considered in the prediction. That's what this ASV based, that is ML, machine learning approach based thing, can visualize, can predict the NMR signal more precisely. So this is the typical H1 NMR signal for zidovudine, and it's from the Gamoxon software system. You can see that, and very clearly, precisely, they shown the signal as well as the integration value. Also, you can see that there is a integration. This red color line that shows the integration value, that shows that speaks about the number of proton in the each signal. And in the other side you can also see that like atom numbers has been given, the chemical shift value is given, and the net intensity as well as the multiplicity also is given. You can see multiplicity and the quality they given, right? You can see this also the quality of the particular signal also they given, like the good, medium, rough. Okay, so this is a way that a prediction tool can use this particular thing. And this graph, this is the thing we developed. We try to establish the regression for the predicted signal versus experimental signal. You can see the correlation coefficient R square value is almost about 0.959. So it is a basic knowledge that remember that if any correlation value between everywhere in science, it is predicted versus experimental or theoretical versus practical. If it is more than 0.9, it is reliable. It is reliable, but still you can see that it is 0.959. It's quite interesting that more reliable five. No, the prediction tool which are available in the NMR, it's almost like similar to the experimental signal value. And this is for the same in C13 signal. You can see that the C13 signal, the predicted versus experimental, the R square value is almost like 0.974. So I mean this the prediction accuracy for C13 NMR is still better than this prediction of proton signal. And now we come for the prediction of UV visible spectra. As you are aware of that, we have Woodward rule. The question is the limitation of this Woodward rule is something that you cannot apply this Woodward rule for all chemical structure. This Woodward-Fisher rule you can apply only for 1,3 butadiene. Then the Scott rule is goes for your alpha, beta, and saturated chemical carbonyl compound. Then you can go for you have the rule for calculating lambda max for polyenes, and you also have a rule for calculating the benzyl compound like a benzene substituent compound of carbonyl compound. But the question is here that if any chemical structure which is falls beyond this category, there is no diene system, the chemical structure. How we are going to predict this UV spectra? How we are going to predict the lambda max? So for that case, you can use this prediction tool, and this the UV spectral prediction tool, and usually most of the things it follows the DFT calculations post approach. They claim that that TD-DFT approach is the most effective approach, and this particular approach it optimizes the geometry file of the chemical structure, and so that chemical structure it's very, I mean, that predict the very accurate results for the UV spectra and lambda max. And so they say that when you go for this DFT fit calculations, and you can use this IQ mode software package, and for optimizing the structure. And this is one model file that when you go for your the TD DFT XRD state for the pseudovidi, because here that how this energy is calculated, that whenever the UV spectrum it's purely based on the molecule which goes from ground state to excited state. But this particular DFT based calculation, it will calculate the energy when your particular chemical structures in the ground state how much, when it goes to excited how much. So what is the difference between these two structure energy? So that energy is converted into lambda. That energy is converted into lambda. Then that will convert it into the probabilities, and based on that, it predicts the lambda max. And this is that one kind of your experiments data we developed, that is experimental versus predicted. So you can see caffeine, we are used solvents, right? In solvent methanol experimentally 272, but predicted is only 258. You can see the percentage error is 4%. But malic acid is 210 and 213, it is the percentage error is only 1. Primidone 258 to 49, 3%. Acetaminophen 254 and 249, it is 1.1%. Benzocaine also we have 5%. And pyrrole is, you can see 210 is the experimental, but predicted is 185. So what I want to say here, And when compared to NMR tool and whatever the existing prediction tool for UV visible, it is not that reliable. You can see the percentage error is up to 5 nanometer. It is fine. So my because what Woodward Fisher rule says, I think, but Woodward Fisher rule claimed that and calculated lambda max by the particular Woodward rule, it is plus or minus 5 with the experimental. That is their climb and their accuracy of the word rule. But this software, you can see that it is oscillating. I mean, this nanometer, even you can see that there is a 11% deviation in the particular difference, especially pi road 210 and 185. There's a lot of difference in the particular predictions. So obviously you can see that lot of outliers, lot of deviations. The particular spectra here, the correlations also it is 0.926. It's relatively low, and still we see the 0.92. It is reliable, but the what computation people and these people they claim nothing but, and even the real experiment, among the real experiment itself, intraday itself, there is a 2% deviations, and whatever the 10% or 12%, they're all they see that it's a kind of considerable and predictions, and so that the R square value also 0.92. So this is the kind of the UV spectrum of zerovidin, and which is generated by the prediction tool is called as Avogadro, and it's all like different maximas 406 and 324 and 279. So lambda max is 279. So usually, and this is a pattern. That's one advantage, and advantage of this Avogadro or the prediction tools are, in case of your in case of your in case of Woodward Fisher rule, just you can calculate the lambda max, but we are not in position to visualize the spectra that which are the wavelength at which the molecule excites more probably, and what are the maximas, how many maximas we get. That and all it is not possible to predict in case of your Woodward Fisher rules. Just a calculation. It is. But in case of this tool, you can see that based on the structure, it also given this. There are three different maxima: 279 and 324 and 406, and out of that, 217 is the lambda max. So you can able to visualize the rough and the UV spectrum for this compound. So here we are coming to IR Raman spectra. Similarly, the same computational tools, the Avogadro, and as a graphical user interface, can also predict the IR spectrum and Raman spectrum for this. So you can see that there is a spectra and of a compound that how a predicted IR spectra is appearing to be. You can see that predicted IR spectra, it's clearly showing that band absorption like 3500 and 3900. There are OH and NH, and you can see 1669, and it's you can see that the predicted value, the frequency, are in decimal here. It's same like your practical also we get in decimal, but here also they can able to predict the particular IR absorption frequency in decimal. So this is a carbonyl compound. Always you know that any compound which shows the IR absorption at 1700, it is a carbonyl compound. It's true, the compound is a carbonyl compound. It's predicting at 1669 almost, right? And you can also see that 1209 to 1097, these are all like C single bond O, all the case. So likewise it's able to, and this side also you can see that various data that can be exported and everything. And so you can see that similarly, like we tried to correlate the predicted data for different compounds like malic acid, primidone, and acetaminophen with the experimental value which we obtained from the laboratory, right? So here, if you look at that, we have taken only just we are done for many compounds, almost more than 50 compounds. We have done it, but I'm showcasing only for 3 compounds just for your understanding. And you see here
For malic acid, we have taken some model frequency, like how the standard and predictions, like deviations, how much it is there, the percentage error. But here also you can see that the percentage error, sir, is a huge here. You can see it minus 10%. You can go around 28% deviations also sometimes there, and sometimes 18% and 29%, all those things. So that's what we feel, nothing but and this same like UV, this IR Raman spectra frequency predictions also, the predicting ability of this particular Avogadro software. What is that? The percentage error is it's varying from even sometimes very accurate. We can also see that if you look at this acetaminophen, that 1690, it is a standard value, and the predicted is 1689. So there is a error is only one. It's a 0.06 percentage error. Similarly, when you go for here, there is one, the pyrimidone, that 2990 centimeter inverse and infrared. This is for CH stretching. But in infrared spectra, it is predicted as 21115. It's it's almost going to see triple bond CRC triple bond H, and so obviously this it is kind of making its particular tool as a limitation. Even it changes the one functional group region into another functional region where the 29% deviation is also available. So that's what I wanted to explain, that when you use this computational tool for IR and Raman spectra also, the caution is required, the validities are not that reliable. And you can see that there are some cases, and here in the scatter plot, what we can understand, nothing but you can see that, and this particular prediction of the frequency, especially regression plot for Raman shift of the predicted experimental, the frequency prediction below 2000 centimeter inverse, they all have very good integrity. I mean, kind of the correlations is very good. I mean, the predicted versus this is a line of regression, and where the predicted versus experimental are on the line. But when the frequencies goes beyond this 3000, especially it goes 1500, we can see the deviation of this prediction versus experimental, that is making your predictability is very poor. So now this we can this is giving you kind of a knowledge that, and the prediction ability of these online tools for the frequency of below 3000 or below 2500 in the fingerprint region of middle IR region may be more reliable than their functional group region. Of course, what I believe is here, what I presume is here, and maybe the predicted frequency may be correct, but this in case of your sometime that experimental maybe sometimes goes wrong, because most of the case, if your sample contains water molecule, that water molecule will enormously affect the frequency of your OH testing as well as NH groups, NH all the case. Okay, that may be the case. There is a lot of deviation in the functional group region. When you come for the fingerprint region, obviously it is fine. Similarly, you can see the same thing. I can see that here completely in case of IR also, the experimental versus predicted, this particular fingerprint region, that is fingerprint region, is comes around up to 1100. Okay, so up to here it's fine, 1100 is also fine. It goes up to here 2000 is fine. So up to 2000 fine, but only after 2000 you can see that the scattering is, the deviation is very high. I mean, between the predicted versus experimental, that is what we observed it. And that's all about your prediction ability and the information about your IR spectra and Raman spectra, a small knowledge for your kind update. And coming to the prediction of chromatographic retention behavior. So when you come for the prediction of chromatographic behavior, and it's very important area, and which is very essential in the present pharma industry, because developing a chromatographic method, it's every day, though the technology is advancing, as well as the chromatograph, the instruments supplied by different vendors are having several features. but still, the method variable selections and the method optimization the hand of analyst. So there is no liquid chromatography equipment is available. Just you give a drug, it will give you a chromatogram. Definitely the need of your intellectual, the need of the human intellectual is very, very essential. So obviously when you go for this chromatographic retention behavior, and so far I'll tell you, I wanted to bring to your notice that till today the analytical methods which are developed by industry people or which are developed by academic researchers are more of OFAT, one factor at a time. We just, it's a kind of conventional approach. We don't study the interaction effect of variables on the retention time. So here we are speaking about retention, nothing but retention time. The chromatography, it is the primary objective of analyte, nothing but, sorry, the primary object of the method is the analyst should retain the molecule on chromatographic column. That is the number one objective. That is very important. If your analyte is not retained on the chromatographic column, then the ultimate, the chromatographic condition is not of useful. So the prime objective of analyst is he has to retain the analyte on chromatographic column. Then he has to elute that retained analyte from the column in symmetry pattern, which acceptable SST parameter, that is system suitability parameter. That's what we called as the theoretical plate should be more than 3000. Tailing factor should be less than 2, or the asymmetric factor should be in between 0.95 to 1.05. And if two drugs are there and both the drugs should have a different retention time, and so that the adequate resolution of more than 2 should be there. That is the basic objective of an analyst when they develop a chromatographic method. But the question is here, and being an analyst, the chemistry of this very important component is lacking in most of the analytical students. And I wanted to stress that most of the pharma analysis students, they think that we are pharmaceutical analysis, we are away from chemistry. That is your ignorance. It's your ignorance. Without your fundamental chemistry, without applying your chemistry of the analyte, or without applying the physical chemistry or organic chemistry into the chromatographic method development, you cannot succeed in their good method, or you cannot succeed a faster development. If you don't incorporate chemistry into your analytical method development, and your approaches are the blind approach, it's very essential that one, you should understand the physical chemical property of analyte. What is the physical property, what is the chemical property of the analyte? Second, the column chemistry. The column is chromatographic column also, it is a full of chemistry. You look at your textbook, it's all chemistry only. It's a bonded, sometimes we call it a C8. Why the C8, why C18? Now I just wanted to bring to notice that there are C30 also coming into the picture. Phenyl column, C30, C24. The column chemistries keep on growing. So until and unless one, you are not able to understand the chemistry of your analyte in the column. So here you have to understand, when your analyte is injected into the column, inside the column, imagine that inside the column, there is a chemistry of column. There is a analyte chemistry. I mean the physical property of analyte, chemical property of analyte. Then there is a mobile phase. Then the mobile phase polarity and this eluting power. This all are the complex things in the particular column that determine your detention of the analyte. So here, the similarly, when you come for your what is that in medicinal chemistry, you know that QSAR, quantitative structure activity relationship, and we also a QSTR, quantitative structure toxicity relationship, and QSPR is there, quantitative structure pharmacokinetic relationship. The similarly in analytical method also it has come into the pictures called as QSRR, quantitative structure retention relationship, and it is the technique is nowadays used for relating the chemical structure of the factor to the retention behavior of analyte in chromatographic elutions. So for the prediction and generally for y variables, we often use as the dependent variable, and x variables are used as independent variables. So here y is it means in case of your any graph or axis, y is we are trying to relate to the chromatographic retention of analyte in minute is called as retention time is y, and x variables are encoded as the molecular structure of the solute. I mean, it's called as encoded as the molecular structure of the solute, I think, but we have so many property for a chemical structure, physical and chemical structure. So all the properties will be encoded as a x variable. And actually, initially, this particular QSRR was used to characterize the column only. So it was used to characterize the column, and by comparing the separation properties quantitatively. But in the day, right, progress, and what happened, this QSRR is developed to provide the information for prediction the retention mechanisms under various chromatographic conditions, especially, as I told you that in a particular stationary phase, C18 is a kind of stationary phase, C8 that is a different stationary phase, and we have C30 different stationary phase. So like a different stationary phase, how we are same drug molecule, how it will behave. So what is the expected retention of this particular analyte on C18 column? What is the expected retention time of this analyte on a particular C8 or C30 column? So it has been extended. And this is, the kind of yeah, that complete flow chart that how a QSRR method is used. So usually the QSRR method what we followed is called as direct comparisons. So it is same that of the person who can do this QSAR, the student who can do QSAR, they can also help you in case of you're doing it QSRR. So you collect the data, you divide the data into true training. One is a training data set, another is a published data set. And you have a training data set, you go for compound name, compound descriptions, and the smile is notations, and we use the file name, right? The structure. What are the chemical structure you're going to use for predicting the molecular descriptor? It should be the smile format. Okay, and then you have a published data set and published retention time of the compounds will be there, and you try to convert into a file, and then you go for try to calculate the system calculations, and then it goes your molecular feature calculations and the electrical feature calculations. So it try to calculate everything, you try to correlate you know training set data and test anything but published data set, right? So it will correlate each other, and it will try to correlate that the molecular property with the retention of your data, because in QSS, yeah, the training nothing but you have to develop a model. For developing model, you have to give a training data set that compound structure, their property, and retention time. The retention time which is already published or which is already available, that you have to give it and you have to put it, right? So altogether, it used to predict your predicted retention time. And just I'll give you an example that how this QSRR predicted retention time and experimental retention time. Here, the experimental retention time, nothing but Remember that the expert detention time is nothing but already which is practically done. We determine the retentions. You can see that here, uh, penciclovir, the predicted is 5.8 minutes. Published is what is the 6.2. And the published nothing but real. I mean, published nothing but what is real in the literature. On the C18 column, the percentage error is 6. And abacavir, it's 5.3 and 5.1. Its error is 5.2. Okay, so here 7.6 and 9.2, and 7.5 and 8.3, and 4.8 and 3.6, 5.6, 3.1, 3.4 and 3. So here, this what it's saying, but the predictions and automatically there are something error is there, that 33% here, right? So here the question is here, what I want to say that this QSR model, it's having the percentage error from 6, 5, 16, 9, and 33% also it's available here. Okay, available here. And the problem here in the particular data, what is showing, but these are the published data. This is the published data prediction time. What we are showing is it is published, but they might have used a different laboratory conditions. But what I want to insist here, nothing but don't think that when you perform your experiment QSSR, you'll have this much high percentage error. It will not. If you develop a model in the same laboratory condition, definitely the retention time prediction and publish will have very closer only, because we have used the published retention time on the particular C18 column only, because the laboratory conditions are different, so that this much variation will be there. So that is the some kind of error from the particular inter-laboratory error also added in here. But when you go for intra-laboratory data, definitely the percentage error will not be this much. That I can assure you that we already done a work on this QS error, and we have published it, and we did not find that much error in case of your predicted as well as published. I wanted to mention that this data is only based on the published data, and we correlated the predicted by using the QSRR. This error is added with the inter-laboratory data also. That is what the thing I wanted to say. And these are nothing but what are the major important physiochemical property or the molecular descriptors that contributing majorly on retention of the particular analyte on the particular C18 column only. Okay. So this regression plot for retention time. So we have taken so many compounds. Just I have shown only few compounds, but almost we have taken more than 25, 30 compounds. QSRR predicted retention time, and we have verified and made a regression plot for the predicted retention time and published retention time. You can see that R square value is relatively good, 0.947, and that shows the model validity, and the way QSRR can significantly contribute the prediction of retention time. Hello, how much time I have?
Sir, half an hour, sir.
Okay, so I should finish by 3 o'clock, right?
Yes, sir.
Okay, then fine.
Okay, sir.
So now I'm coming to this, the chemometrics. So actually, the chemometrics in 1 and 2 dimensional chromatography, and it is the still most of the analytical chemists feel that the chemometrics is the appreciable tool in chromatography, because it is appreciable tool for pre and post data analysis to resolve baseline drift. Because the chromatography, the baseline drift, it's one of the problem or a trouble where an analyst always should focus, especially when we go for gradient. The gradient, obviously, when you go for gradient, when you change the organic or aqueous phase over the period of time, there is a change in the composition, change in the absorbance, change in the specific gravity or density or viscosity of the analyte. Obviously, that changes your absolute detection behavior or detection characteristics of your mobile phase. And also it is used for undesired background signals, and also to predict or assess the shifting of retention time. Sometime the retention time of a drug will shift from interday or intraday, or from for each and every injection also sometime the retention time will shift for 0.1 or 0.2 minutes. And also there are sometimes if you have a related, especially in case of your chromatography, related substance, as you are aware of that, related substance are structurally very close to the analyte. So if your mobile phase and columns are not that appropriate, it cannot resolve it. Or sometime there is a small difference in your impurity and your drug, that may not the Lockwood difference between your drug and impurities are very narrow. When the Lockwood difference are very narrow and both will have similar lipophilic character, it will have a similar retention behavior and both will correlate together. So that will become the unresolved peak. So usually that many analytical chemists or the computational analysts, they use this chemometric method to resolve this resolve or solve this unresolved peak in chromatography, or the shifting retention time in a chromatography from interday or intraday, or undesired background signals, or this baseline drift. Everything they have used it. And further in industry also, some of the industry they used, and they have a raw method, nothing but the method which is conventionally developed. They already have, with the trial and error, they developed a method. So what the industry, sometimes the chemists, what they do, have adequate knowledge in chemometrics, and they try to improve that method performance, like SST parameters. For example, I have the particular drug is eluting, that eluting resolution is only 2. So now when your resolution in a particular method is a 2, and there is a possibility when you go for method transfer, and method transfer from the analytical R&D to quality control unit, there is a possibility that there will be a reduction in the resolution also. If there is a change in the retention of any one drug, a first drug or second drug, automatically what happened? The resolution will come down. If resolution come down to 2, automatically it fails. SST will fail. System suitability, I mean USP SST, that is USP SST will fail. So if it get fails, for that what happened? Obviously that's what, when USP prescribe that the resolution between two peaks would be 2, and we always try to develop a method that resolution at least 3. So now in such case what happened? The analyst, he'll try to increase the theoretical plate by using chemometric method. When they try to increase the theoretical plate, nothing but peak sharpening. When the peak sharpening is there, the peak width will come down. It means when you say that my peak theoretical plate is very high, it means your peak is very sharp and the peak width is less. So such a way, the analytical chemist, they'll try to improve the existing method, the existing raw or conventional method, by using chemometry towards better performance. And there are different chemometric approaches used in the chromatography, and that I'm not going to discuss in detail. These are the computational things. Only the statistical people can tell you better way. And there are different approaches, like PLS, we called as penalized least square, or sometimes it's called as MC, or called as multivariate curve resolutions, orthogonal subspace projection for background corrections, local minimum value approach, baseline estimation and denoising using scarcity, retention time alignment strategy, and also we have peak clustering and principal component analysis. Out of these two, you just forgot about being analyst and you always try to have experience of using this PLS. Number one, penalized least square method. The last one we call as PCA, the principal component analysis. Now this principal component analysis, very very important case of your metabolomics and proteomics studies. And now in the current chromatography techniques like LCHRMS. So in Naipur Hajipur, we have LCHRMS. In our analytical department, we have LCHRMS that cost about 5 crores. So for this LCHRMS, we can buy two LCMSMS quadrupole. So that now we are using this LCHRMS especially for the metabolomic studies and metabolomic studies and proteomic studies, where we are trying to relate the clinical samples, biological sample or pharmacology sample. We are trying to study which is upregulated, downregulated, and that is relating to the biomarker identifications and study of toxicity mechanism. These are things we started doing it, but in that especially, and we have the software which is inbuilt in the particular LCHRMS compound discoverer or proteome discoverer, that is purely functioning on this PCA only, principal component analysis. That principal component analysis are trying to match the control and diseased, and which is upregulated, which is downregulated. It is upregulated, how many fold is high, how many fold is low. So the even that is correlated by part and parcel of a chemometric approach in metabolomics and proteomics data interpretations. And usually, and in chemometrics, it is just for a fundamental thing. Just touching the point that there are in chemometric there are supervised and unsupervised techniques. Usually, if it is an unsupervised approach, as I mentioned, the PCA, the independent component analysis ICA, and cluster analysis can be used to understand the dissimilarity variance in the data matrix. But in case of supervised techniques, and can you please mute the mic? Somebody is disturbing.
I request all the participants, please mute yourselves. Please switch off your mic.
Yeah,
Okay, sir.
yeah, thank you. And in case of unsupervised techniques, and such as the most recommended will be the multivariate calibration methods, and can be used, and especially when data does not cluster against any objective criterion. That's very important that you can use. Even it doesn't matter that you can also use this PCA principal component analysis also can be used in case of supervised. That's what I said. You use PCA. PCA can be used anywhere else, not a problem at all. But especially this multivariate calibration methods are most recommended in the chemometric methods only when the data does not cluster against any objective criteria. And that's what here I just wanted to mention that the PCA and multivariate calibration methods and PLS, all things. Now, finally, coming to this analytical quality by design. So, when I speak this analytical quality by design, it is one of the very very interesting area in pharma industry as of now, because FDA is insisting or ICH is insisting to implement analytical quality by design in line with the product QBD. So here, this is a kind of hybrid computational approach. Obviously, it is not a prediction. It is not a prediction tool. It is a hybrid approach where you have to hybrid your computational approach and the experimental approach to bring out a robust analytical method. That's what I wanted to say. This analytical quality by design. So generally, the concept of QBD is applied to the analytical method. We call it as analytical quality by designs. So what are the advantages of this analytical quality by designs? And these analytical quality by designs allows the pharma industry or analyst to change the method variable within the method operable design region we called as MODR. This MODR nothing but design space. And this, if any analytical method is developed by using this AQPD approach that gives high degree of robustness, you can also orient towards developing a cost-effective method. It provide better scientific understanding between the critical method variables and method response. Also provide a regulatory flexibility in filings. It is also reduce the risk of method failure during method transfer. At this point, I just wanted to insist here. In pharma industry, as of now, this method failure during method transfer is very common. It means a method is developed in an analytical R&D. The developed method is validated. The method parameter passes the validation criteria of ICH Q2, but the same method when it's transferred to the site, quality control department or any QC unit, the same configurations and same method conditions, sometimes it fails. The reason nothing but the lack of robustness, and we don't develop a method with a better scientific understanding among the variables and method response. That is the problem. So ultimately, the method which we are developing now conventionally, of that is the reason why we transferred the analytical method from R&D to site. I mean the industry, or you transfer your R&D to QC unit, and they are, even though you transfer, even though you validate in your R&D side, you have to revalidate it. I mean you need to ensure it. So here, this when you use the analytical QBD, you can avoid such things. And another for your audience, scholars, now pharma industry speaking about Six Sigma. Six Sigma. What is the Six Sigma? So the Six Sigma is a statistical term that represent that they believe that if any pharmaceutical process or manufacturing or testing is designed based on Six Sigma, that process will have 100% success. I mean, the risk of failure is 0%. So that's why here the similar concept, if AQBD is the tool for attaining Six Sigma. So if you use Six Sigma in analytical method, this will eliminate the out of trend result we called as a WOOT. It may also eliminate out of specification WOOs, out of control WOOC, and out of statistic control WOOs. So here, that's what here the strength of the concept is. As of now, the industry is right analytics at right time. So that eliminate your failures and it makes 100% success. There is no method failure. Why this analytical QBD should be during your product QBD? So these analytical methods, whomever the case, yeah, manufacturing or in process, anyway, raw material analysis. So these analytical methods are the indicator of the quality of the product. Without analytical method, it is very difficult to assess the efficacy and quality of the product. How without analytical method your product is quality or efficacy? And the analytical method is the part and parcel of your complete product life cycle. You are aware of that. It starts from raw material, it ends with once upon a time we used to call that analytical method ends with your finished product. No, now the analytical method is part of a real-time testing, real-time stability testing. And it is the analytical method is the one that is the indicator of the relationship between your process and product. How you, without the analytical method, how you are going to say that the blending uniformity, or we called as BU and CU, content uniformity, blend uniformity, and everything, right? How you are going to say, until and unless you determine the content, you cannot relate your process and the product quality. So this is your for your kind knowledge is preferably perhaps and the first review article on analytical quality by design in India. And this is, I just for your kind uh, knowledge that how this analytical quality by design approach is done in computational, where and all we use this computational approach in analytical quality by design and how you process it. So before you going for your, analytic quality by design approach, and you should understand generally we always called as analytical method performance. And what are the indicators of analytical method performance? And always the term we pronounce every time: accuracy, precision, specificity, limit of detection, limit of quantification. All those terminologies are nothing but method performance indicators, or you can call it as method performance parameter. So now how you are going to measure the method performance? If your method is accurate and precise, your method performance good. If your method is not accurate, or if your method is imprecise, then your method is performance is bad. That's it. So we call it as analytical target profile. See, if you look at this analytical target profile, and always the analytical quality by design approach, it starts with the analytical target profile. So what is that analytical target profile? And being an analyst, you should ensure what are you going to analyze, where are you going to analyze, when are you going to analyze. You try to address these three questions. What? I am going to analyze paracetamol. Where? In tablet. Where? During in process quality control, in stability assay, or in uh, finished product. So this is going to speak about that analytical target profile. That's why here analytical target profile either assay or it can be impurity. If your stability, you want to find your impurity also. You're going to identify it, everything, right? So here, based on that, you define your method, you select your method, you define your method performance criteria, how your method should be there to your target profile. Okay, your method should be specific, your method specific should be accurate, method will be precise, and all the things you are to define, and you are to justify that the analytical methods selected is going to justify your analytical target profile. Then you go for your risk assessment. So the risk assessment, nothing but that when you go for risk assessment, you are going to consider all parameters like instrument parameter, drug related parameter, and your method related parameter. All the parameter you're going to list out, and you're going to link scientifically, logically to the method response. How it's going to affect your method response in a particular instrument. So that's why here we have a different types of risk assessment tools like FMEA and Pareto analysis and Fishbone. Everything, we have so many things are available that we can go through in the web. It's available widely, right? Then you go for your method development validations, and in that you consider your DOE, the design of experiment, and you construct design space that is called as MODR. Okay, then once you, if you bring out a design space, you can able to define your control strategy, and you can define your control space, and you can meet the method performance criteria in that. And also then you can define your continual improvement. That's what here, the things are, completely from technique selection to continuous improvement. It's all like, and once again, there is a problem with your method when you monitor your method performance. Once again, go back to the technique selection, then you start doing it. That's why it's called as continuous improvement. And this is a slide I just wanted to showcase, like, how this is like comparison between your AQBD and analytical QBD. The same, right? In AQBD, you go for your analytical target profile, do risk assessment. When you do a risk assessment, critical, critical method attributes, your defined key method parameters. This you all consider this all. This is a DOE, right? So here, why I brought this AQBD in this computational approach, you can see that the risk assessment to method operable design region, it's purely depends upon the DOE and statistical analysis. The DOE nothing but it's a kind of your artificial intelligence and computational approach. You are going to be a part of your AQBD. So that's what I hear. Nothing but whatever things here, and everything is a hybrid of your computational approach and your human brain. So the finally the AQBD nothing but it's a hybrid approach of your human intellectual and artificial intelligence. Human intellectual and artificial intelligence both has to combine in order to do this AQBD, then the expected success will be 100%. What is this analytical target profile? And it's just for your knowledge that, and as I told you, the method performance characteristics are accuracy, precision, and specificity. And usually the accuracy, specificity, linearity is coming under systematic variability. Precision, detection limit, and quantification limit comes under inherent random variabilities. But range and robustness, we don't not applicable. Why this is mentioned here? I think, but see, the very important is specificity and linearity. So if your method is not specific, definitely it will not be a linear. If it not linear, it will not be accurate, or it will not be precise. So that if you test your accuracy precision, people we feel that it is by default the method is the linear, and what is the specific kind of a logic what we use it. So the last, it is called as always I was speaking about MODR, method operable design region, and it is the kind of contour plot we call it as a response. It's a kind of a response surface plot. You can see that how you what is the design space here. You can see the yellow color space, and you can see this pink. The pink color nothing but where you can the retention time of the drug is more than 6 minutes, right? More than 6 minutes. For example, now you wanted to have your retention of the drug is more than 6 minutes. Okay, and you can fix those parameter here. The x3 nothing but this particular at a rate of flow rate is 1.2 ml. You can fix your flow rate at 1.2, and x-axis is pH and y-axis is percentage aqueous. So you can see that, and here x-axis pH, you can see the complete pink color throughout the axis. So in these conditions where the retention, if your retention time is more than 6, it is a pH independent, right? The pH has no effect on your retention time, because wide range, because -2 to +2, almost like if you encode it, the pH ranges from low to high. Even if you change any pH, it is not affecting your retention time after 6 minutes. We come to next. You can see this will give you better, right? Now I wanted to, for this contour plot what we developed, I wanted to have my retention on in between 5 to 6, in very narrow range, 5 to 6. You can see the green color here. Green color, you have very good space for the green color that indicates a design space for where you can able to elute your drag at 5 to 6 minutes, 5 to 6 minutes, and where the x2, that percentage aqueous in the mobile phase is 15%, and pH is x-axis, and flow rate is what is y-axis. The flow rate is y-axis thing, but it's the domain 0 level, and it is go +2 level, and almost like if you take this domain, if this range to this range, I think, but almost like the flow rate is 1 to 1.2 ml. Even the flow rate from 1 ml or 1.2 ml, and even your pH is almost like at this range is 6 to 7, or automatically what happened, and syntax that indicates blindly. If for to understand that you have raw data, that raw data I not disclose here, but still the space speaks about that we have wider possibility that we have greater space for getting this drug retention 5 to 6. So here for this condition you can see that there is a see white color here, white color 4.5 to 5. So at this condition of X2, 15% mobile phase, 15% is aqueous in mobile phase, and at this given pH, at the given flow rate, and it is very difficult to retain a drug in 4 to 5 or 4 to 5. So you can see that there is no color in this particular design phase. There is no yellow color, there is no red color, there is no white color. Indicates, and this particular set of experiment do not provide provision to elute a drug and below 5 minutes. It is possible only from 5 to 6 minutes. Next is what is the 6 to above 6 minutes. So that is the kind of understanding we try to get from this particular AQBD and contour. We call as MODR. We call as MODR regions. So conclusions. Finally, I want to conclude that the computational approaches in prediction of analyte behavior can provide the pinpoint accuracy and the smallest possible of deviations. Always remember that the prediction always not final. It can be initial assessment to validate results in that defined window. That's it. And it can be useful in the resource limited setup as we are in the era of computer and internet. And computational analytical behavior prediction has numerous applications in the academic research. And I expect all the scholars and teachers explore your internet as you all the time internet is with you. You find out the possible online tools can be used for the computational use and predictions. So now finally, I would like to recommend all the scholars to equip the skills on the computational predictions with respect to your field. With this, I thank VVIPS organizers and AICT, ISTE for the provided opportunity to deliver the talk on this computational approach in the prediction of analytical behaviors in chromatography as well as spectroscopy. Thank you very much.
Thank you, sir. You have explained new prediction tools for spectral interpretation and also various computational approaches for analytical method development, and also explained a detailed explanation of the AQPD applications. Thank you very much, sir. I request all the participants, if you have any queries, please either you can unmute yourselves or you can post the questions in chat box.
Good afternoon, sir.
Yeah, good afternoon, please.
Sir, is it possible or is it feasible to do this type of courses in UG level, sir?
Yeah, it is possible,
Like introducing.
No. What I see is UG level, the students are UG, but the present
What I
generation.
mean to say is it feasible for planning this type of experiments?
Yeah. I'm answering it. Am I audible?
Yes, sir, you are audible.
Yes, sir, you are audible.
What I want to say is the students are UG, but your UG students are masters in computers.
Okay.
Definitely, you can orient them. You can orient them. They can do better way that you just give you a way that you just guide them. These are the tools available. Try to explore the methodology, procedure. Everything is available in the internet, the manual itself. They can do it. They can do it. Yes, that's what I'm saying. This computational approach is more reliable for these generations. But always guide them that it is not an endpoint. It is only prediction. It is not true. It is only 80% true. That is it.
Okay, sir. Okay, sir. Thank you, sir.
Okay, sir. Thank you very much. On behalf of PV Institute of Pharmaceutical, it's my privilege and honor to present you e-certificate, sir.
Okay. Thank you.
Please accept it, sir.
Okay. Thank you very much.
Okay, sir. Thank you very much, sir, for your elaborate explanation of the new computational approaches in analytical method development. Thank you, sir. I request all the participants, please find the feedback link and assignment link in the chat box. Once again, thank you to all the participants and speaker. Dhatri?
Okay. Thank you very much. Can I leave?
Sir, okay, sir.
Thank you very much. Thank you very much.
Thank you, sir.